Hi everyone, Phil Wong here, and welcome to your lecture on skeletal muscle. Now, the placement of this lecture within this course is strategic because remember, as you recall, a few lectures back, we introduced the four major types of tissues, and we talked about epithelium and connective tissue, and then we said that muscle tissue and nervous tissue will follow a little bit later. And then in our prior lecture, we were discussing joints and synovial joints in particular, and we discussed how skeletal muscles are attaching and anchoring to bones, and when they contract and shorten, they're going to cause movement at these particular joints. So again, it makes intuitive sense that we're going to move on and we're going to talk a little bit about the tissue that comprises skeletal muscles. Okay, so this is going to be your lecture on skeletal muscle. Have a look at some of the objectives over here. So what we're going to do, first of all, is provide a broad overview of the three types of muscle tissue, how they are similar, how they're different. So we'll look at skeletal, cardiac, and smooth muscle tissue. And what we're going to do is really focus today on skeletal muscle. And then what we'll do is that when we start talking about the heart and start talking about all these different organs that contain smooth muscle tissue, then we'll kind of come back and we'll discuss the finer details of those particular types of muscle tissue. All right. So for skeletal muscle, we'll start off kind of a broad overview of the tissue, how it's comprised. We'll talk about how the skeletal myocytes are packaged within layers of connective tissue. So again, there's a very precise organization of the myocytes and the connective tissue packaging. And then we'll spend the rest of our lecture going into detail about the histology of the skeletal myocyte. We'll talk about how you have all of these structures that are in common with all the other cells in our body, but again, these cells have a different shape. They have different structures and organelles and contents within them that are responsible for responding to some sort of electrical stimulus. Okay, and from there, these cells are going to link that electrical stimulus to a contraction event. Okay, so this is what we refer to as excitation-contraction coupling. So I'm going to be going over that premise and that theme again and again at different points in the lecture. Just be aware, in this lecture, I'm going to be moving back and forth between the PowerPoint slides and my whiteboard because I think the diagrams are pretty good in what's included in this lecture package. But for me, in particular, when I'm trying to visualize how these structures kind of come together in the myocyte, that's why I kind of like to draw things out. So I'm going to switch back and forth so you kind of have both experiences. All right, here we go. Skeletal muscle. All right, so let's talk about muscle tissue as a whole in terms of what it's comprised of. So first of all, when we talk about the cells that comprise muscle tissue, the majority of these cells are going to be myocytes. Okay, so another name for a myocyte is going to be a muscle fiber or a myofiber. So I tend to interchangeably use these terms. Okay, so just be aware these cells are known as the myocytes or the muscle fibers. These cells are, they contain your average cell organelles. They have a plasma membrane that kind of surrounds it and separates it from the extracellular environment. But there are some very important properties of these myocytes. So first of all, they have the ability to respond to an electrical stimulus, and as well, they are able to conduct their own electrical signal. So again, this signal is what we refer to as an action potential. So when I'm talking about a specific myocyte, I will largely refer to it as, if we're talking about a skeletal myocyte, I'm going to talk about the skeletal myocyte action potential. Okay, so that's the first thing, is that we can that these cells respond to an electrical signal, and then they can generate this propagating electrical signal of their own. Okay, and then what's going to happen is that these cells also have the ability to create a tension force. So you have structures within these myocytes that are able to contract and shorten in response to this electrical signal, and they're going to create a tension force. Okay, so there are three types of myocytes and muscle tissue. We have the skeletal, we have the cardiac, and the smooth. Okay, so let's have a look just right now at the cells that comprise skeletal muscle. So first of all, these skeletal myocytes are going to be very long cylindrical cells that are going to be arranged in parallel. Okay, these are one of the few cells in the body that are multinucleated. So they have multiple nuclei per myocyte. Okay, so that's what they're showing you over here. So this is going to be your diagram on the left and your actual histology slide over here. And what they're trying to show you, so these are going to be small little sections of individual myocytes. So these are skeletal myocytes over here. And we're noting that we have multiple nuclei per myocyte, okay? And that's kind of demonstrated in the slide on the right. It's a little bit hard to note, but if you kind of take maybe this one over here, so if this is a skeletal myocyte over here, you can definitely see here's one nucleus over here, here's another one over here. Okay, so they're multinucleated. I also want you to pay attention to where these nuclei are located. Do you see how they've drawn them at the periphery of the cell on the side? Okay, so you can see they're on the side as well. Okay, none of them are really in the middle of a given myocyte. And the reason for that is because most of, and the majority of the inside of a myocyte is taken up by these very long protein threads, and we call them myofibrils. Okay, and the myofibrils are going to contain those sarcomeres, which we're going to talk about. But all these myofibrils are what's going to provide this property of contracting and shortening in the myocyte. I also wanted to kind of delineate the fact that skeletal muscle, okay, and skeletal myocytes are voluntary and they're under conscious control. Okay, so what's going to happen is that we have innervation from the central nervous system, and then we have a variety of motor neurons that are sent out to these skeletal muscles. Okay, and so it's a very conscious decision if we are going to contract these muscles or not. Okay, so that's what we refer to as voluntary. Okay, so that's skeletal myocytes. We're going to come back to that, but we're going to just introduce some of the other muscle types. Okay, so if we move to the next slide, we are now talking about cardiac muscle, which is going to consist of cardiac myocytes. Okay, so these are going to be very similarly structured cells, but there are some very specific differences. Okay, so first of all, the main difference is that, while we have these very long cylindrical skeletal myocytes, these cardiac myocytes are actually very, very short, okay, and they exhibit a branched pattern, okay? So you can see you have a branched pattern over here. We're looking at these skeletal myocytes, okay? And again, they're short and they exhibit a branched pattern, and you see what they're going to do is that they're going to connect up with neighboring cardiac myocytes. Okay, so a branched fibers. Okay, and as well, the point of contact between neighboring cardiac myocytes are going to be these very discrete locations, which we call the intercalated discs. So these are specialized cell junctions that are only found in cardiac myocytes. Okay, and they tend to be a lot darker in the histology slide. So if you're looking over here, these are cardiac myocytes over here, and these very, very dark bands over here, these are going to be your intercalated discs. I can see another one over here, okay? Maybe another one over here, okay? And so that is contrasted with the striations. See this alternating pattern of light and dark bands, okay? That's associated with cardiac myocytes. And if we flip back over here, we also noted that skeletal myocytes, again, they are multinucleated, but they also exhibit this striated pattern. Okay, so again, we have these light and dark bands. Okay, and so this is going to be because all of these myofibrils are lined up in a certain way. Okay, and that means that all their sarcomeres are lined up. So therefore, we are going to get this light and dark pattern. Okay, so more on that to come. So you see these cardiac myocytes and these skeletal myocytes, they share these certain common features. Okay, but at the same time, there are some differences. Another difference in the cardiac myocyte is that this is a mononucleated cell. Okay, so there's one nucleus per cardiac myocyte, and we also consider cardiac myocytes to be involuntary, so they're under unconscious control. So we can't consciously demand that our cardiac muscle contract and relax as required compared to a skeletal myocyte. Okay. And lastly, we talk about smooth muscle, okay? And smooth muscle is distributed throughout a variety of different anatomical structures, okay? They're in the walls of your structures of your GI tract. They're in the walls of your larger blood vessels, okay? And so when we kind of look at smooth muscle, these cells look a little bit different. So first of all, they tend to be spindle-shaped, okay? That's kind of best exemplified in this illustration over here. And when we talk about spindle shape, we're referring to the fact that these are very elongated cells with a very thick middle and then a tapering at the end. Okay, so they're showing over here, they're showing kind of this spindle-shaped cell over here. Okay, there's, these smooth myocytes are mononucleated, okay, a centrally located nucleus over here, and there are no striations, okay, and that's in comparison to the striated pattern of the skeletal and the cardiac myocytes. Now, this histology image isn't kind of the best to kind of exemplify, the actual shape of these smooth myocytes. Okay, so you can't really see the spindle shape aspect of it, but you're kind of looking at the consistency. You can definitely see like these are all smooth myocytes over here, but we don't see that same light and dark banding pattern that we saw in the previous slides. Okay, and again, smooth myocytes, smooth muscle is also considered involuntary and under unconscious control. Okay. So let's look and return back to skeletal muscle and talk about these myocytes again, as well as the connective tissue packaging that organizes these myocytes. So again, we have previously indicated that skeletal myocytes are multinucleated, so they have multiple nuclei per cell. Okay, so they're showing you this is going to be a light micrograph over here of a portion of a single skeletal myocyte, a single skeletal muscle fiber. Okay, so you can see that we have previously established that we have multiple nuclei. Okay, and as well, these nuclei are pushed off to the side because we have lots of myofibrils. Okay, that are going to take up the bulk of the inside of this myocyte. Okay, and so by aligning all of these myofibrils together, this is why we get that banding pattern, that striated appearance of light and dark. Okay, and actually, what they're going to do, we'll come back to this image again. But what they've done over here is that, we can see, the myocyte under a light micrograph, okay, stained in a certain capacity. But if we want to even look even further and kind of zoom in, we have to use another form of microscopy. Okay, so this is going to be electron microscopy. And we kind of zoom in even further, and you can definitely see these are going to be the alternating dark and light bands. Okay. So we have multinucleated cells. They have a striated appearance. Okay. And again, what's going to happen is that when we are facilitating the contraction and the shortening of these myocytes, this requires energy. So what has to happen is that we need to have a supply of ATP, okay, in order to use the necessary energy to generate that tensile force. Okay, so just kind of keep that in mind that we're going to need lots of ATP in order to make this happen. And so the majority of skeletal muscle tissue is going to be comprised of these myocytes. But again, what's going to happen is that we organize them by packaging them in these layers of connective tissue. Okay, so that's what we're going to show you next. Okay, so when we're talking about the connective tissue packaging, we're also talking about the ways in which we organize the components of the skeletal muscle. So first of all, we have an overlying connective tissue packaging that is going to cover and surround the entire muscle, and we refer to that as the epimysium. Okay, so this is the epimysium over here. Okay, so it's a layer of connective tissue that is going to kind of surround the entire muscle. And when we kind of look closer, what we would see as well is that within a given muscle, we have these very discrete bundles of muscle fibers. Okay, those are going to be your myocytes. And so those bundles of muscle fibers are what we refer to as fascicles. Okay, and each and every one of these fascicles, okay, is going to be surrounded by its own layer of connective tissue. Okay, and we refer to that as perimysium. Okay. And even further to that, within our fascicle, which is our collection, our bundle of myocytes, okay, what's going to happen is that each and every one of those myocytes is going to be surrounded in its own connective tissue packaging, and that's what we call the endomysium. Okay, so there's a very discrete organization of all of these bundles of myocytes. And again, this also provides a bit of a strategic advantage too, because remember, each and every one of these myocytes, okay, within a muscle requires a blood supply, and it also requires innervation. Okay, so the thing is that remember we've already previously talked about how any sort of connective tissue, okay, cartilage being the exception, the majority of connective tissue is going to be very well vascularized and innervated. Okay, so the thing is that all of our very tiny blood vessels and all of our tiny neurons, those are actually going to pass through the different layers of of connective tissue in order to find their way to each and every one of those myocytes. Okay, so what we've established, okay, is that we have all of these different, so not the different, but we have all of these skeletal myocytes, okay, that are comprising a skeletal muscle, and they are packaged within layers of connective tissue, okay? And now what we're going to do is go into detail about these particular skeletomyocytes and their structure. Okay, so we're going to be coming back to this diagram quite a bit. But what's happening is, sometimes I kind of look at this and it takes me a while to unpack the different components of it. So what I find is easier is that I'm actually going to switch over to the whiteboard and start to draw out this myocyte in my own way. Okay, and so what's going to happen is that I'm going to start talking about some of these structures that are going to be in the next couple slides. Okay, so hold on, I'm just going to kind of flip over. We're looking at a skeletal myocyte. So I'm just going to draw that at the top. So this is going to be your skeletal myocyte. And this particular illustration should only take about 1 page. Okay, you can do it in landscape or portrait, whatever you wish. Okay, but really what I'm going to be drawing is one very, very large skeletal myocyte. Okay. All right, so let's just introduce the major structures. So we noted that skeletal myocytes are very, very long cylindrical cells. Okay, so what I'm going to do is I'm just going to draw one huge long cylindrical cell like so. Okay, so I'm going to draw it like this. Okay, go. I'm just going to this. I'm even going to attempt to draw it in 3D. It's going to be a disaster, but let's go for it. So we are going to draw a cylindrical cell like so. Okay. And we're going to draw one just like this. Okay. Okay. So this is going to be our skeletomyocyte. We have a couple of specific names for structures that we are already aware of. So first of all, the plasma membrane of a myocyte is what we refer to as a sarcolemma. Okay, so I'm just going to write that at the top over here. This is going to be the sarcolemma. Okay, this is the plasma membrane. Okay, right here, fantastic. Okay. As well, there's a special name for the cytoplasm of a myocyte, and we refer to that as the sarcoplasm. Okay, so I'm going to do, here's the sarcoplasm inside. Okay, this is the cytoplasm of a myocyte. Fantastic. And there are some important myocyte-specific features within the sarcoplasm. So we'll talk about that a little bit later, but I'm going to introduce the following. Okay. So I'm just going to write them down over here. We're going to have glycogen. Okay. We're going to have myoglobin. And we're also going to have creatine phosphate. Okay. Okay. So there's our sarcolemma and then our sarcoplasm. Now what's going to happen, okay, is we need to kind of discuss what's going on with this sarcolemma. Okay. So actually, I'm going to do something really horrible, okay, which you're not going to like. You might need some whiteout over here, okay, but I'm actually going to draw this top portion over here into 3 different portions. I'm going to explain why in just a second. Okay, so sorry about that. Sorry about that. You might need some whiteout. Okay. Okay, there you go. And what's going to happen is the sarcolemma, okay, which is going to be the plasma membrane that surrounds your myocyte, your skeletomyocyte, there are going to be some very deep invaginations of the sarcolemma like so. So I'm actually just going to draw it like this, okay? Okay. And we have a special name for those, and we refer to them as the transverse or the T-tubules, okay? So this is going to be the transverse, okay, T-tubules. Okay. So again, these are just invaginations of the sarcolemma, okay? And so what happens is that they're going to invest really deep into the skeletal myocyte, okay? And as a result, what's going to happen is that some of the outlining, so all the cytoplasm, or sorry, the extracellular fluid over here, so this is all ECF over here, okay? so extracellular fluid, okay, that means that deep within the myocyte you also have extracellular fluid over here, okay? I'm just going to erase that over here, but just kind of be aware that, you know, even deep within the cell, deep within these transverse T-tubules, there is some extracellular fluid, okay? So these T-tubules are invaginations of the sarcolemma, okay? Now, remember, we have the major feature of our skeletomyocyte is the presence of these myofibrils, right? These are collections of protein fibers. This is going to be your actin and myosin. I'm going to draw them in red, okay? And I'm just going to denote them as these very, very long threads. So I'm just going to even draw this very long one over here, okay? I'm going to draw a couple of them. I'm just going to draw 3 of them, okay? Go. Here's the third one. Nice, okay. And so those are going to be our myofibrils, okay? So I'm just going to draw that in. This is going to be your myofibrils, and they're going to extend the length of the myocyte, okay? And the myofibrils, okay, if we kind of extend that, these consist of overlapping actin and myosin myofilaments. Actin and myosin myofilaments. Okay. So I can even draw that down over here. Okay. So maybe what we'll do is I'm going to take a small little box, a small little portion of one particular myofibril, like so. Okay. And I'm going to kind of expand it out over here. Okay. So that what we can see is the overlapping actin and myosin. So what I'm going to do is I'm just going to kind of try and keep the colors consistent if I can. But what I'm going to do is I'm going to draw kind of several myosin myofilaments. Okay. Okay. Maybe I'll just draw a couple over here. Nice. Okay. And then overlapping these, you have your actin myofilaments. I'm just going to arbitrarily choose blue over here. Okay. So I'm just going to kind of draw some actin overlapping like so. There we go. And they're going to kind of be linked together. Fantastic. And I'm also going to draw the overlapping nature of your actin over here. Okay. And then they're connected like so. Okay, so a little bit of drawing over here. Okay, not perfect, but that's okay. Nice. Okay, so let's just put a label to that. So this is going to be actin over here. So everywhere you see blue is going to be actin, and then these are going to be myosin over here. And so what you can see is that really these myofibrils, these are consisting of overlapping actin and myosin myofilaments, okay, and they're kind of overlapping each other, and then they're extending end to end all the way along the length of your myofibril. So we're kind of looking at a small little box, okay, but just kind of picture that, you know, end to end for your myofibrils, you just have actin, myosin, actin, myosin, actin, myosin, okay, collections of these myofilaments, okay. And then just kind of to add to that, sorry, we don't want to get too ahead of ourselves over here, is again, I showed you that we have 3 myofibrils, okay, but again, there are going to be tons of them inside our skeletomyocyte, okay, but that means that if you have all these myofibrils themselves that are kind of aligned very close to each other, okay, so what I'm going to do is I'm going to just kind of do a little bit of a cut and paste over here, okay, so what I'm going to do is I'm just going to kind of draw this one over here, and I'm going to copy, okay, and paste. Okay, that didn't work. Let's delete that one again. Let's try one more time. Okay, so let's copy. Let's just do paste. That's what I wanted. Okay, and I'm just going to kind of move it. I'm going to kind of align it exactly like so. Okay, I'm even going to do it again. I'm going to try it. I'm going to copy it again. Let's paste it again. Okay, and there we go. Okay, so what I want you to consider now, okay, is that because we are aligning all of these myofibrils in a certain way, what's going to happen is that now we are going to get these light and dark bands over here. Okay, so again, what's going to happen is that you're going to end up, because all the myofibrils and all of these myofilaments are aligned with each other, this is going to be your A band, okay, and this is going to be the dark band in your striated pattern, okay. And you're also going to get this light band over here, okay? And that's going to be your I band. And that's going to be the light staining portion that you see in your striated pattern, okay? So this is kind of a food for thought that's going to come back. This whole idea that that's the reason why you get your striations, okay? But again, for the time being, remember, let's come back to what we know already, okay, that we have our sarcolemma. The sarcolemma is going to invaginate down into these T tubules, okay? And as well, what's going to happen is that you also have the sarcoplasm, which is going to contain all of the normal cytoplasmic components, but in addition, we'll have glycogen, myoglobin, and creatine phosphate, okay? And then, of course, you know, the major player is going to be your myofibrils. So tons of myofibrils in the middle, but again, what's going to happen is, remember, you have your nuclei that are pushed off to the side. So maybe we'll just draw a nucleus over here. So let's draw a nucleus over here. And remember, there are going to be multiple nuclei over here. Okay, so this is going to be your nucleus. Nucleus and your nucleus. Nice. Okay. One other thing I want to introduce is that these myofibrils, okay, they're very densely packed into the myocyte, but they're also surrounded in specialized smooth endoplasmic reticulum, and we call this the sarcoplasmic reticulum. So I'm going to draw this in purple over here, okay? And so what's going to happen is that this endoplasmic reticulum, the sarcoplasmic reticulum, okay, is going to surround these myofibrils. So what I'm going to do is, yeah, let me just do it this way. So let's, I'm going to draw them. They're kind of like, if you kind of picture them, they kind of function like water wings that are kind of surrounding the myofibrils. So let me just draw it that way. They're kind of like cylinders like so. They're kind of like those water wings, okay? And what they're going to do is that they're going to kind of surround regions of the myofibril, okay? Okay. Like that, okay? Let's just draw a couple more over here. Okay. And one more on the right. Okay, so they're kind of like surrounding these portions of the myofibril, just like a water wing. Okay, so these are going to be, so let me just label them in over here. So this is going to be your sarcoplasmic reticulum. Okay, so this is the sarcoplasmic reticulum. Okay, and what's going to happen is that they are significant because they are going to contain calcium. Okay, so contain calcium. Okay, that's going to be your calcium store. Okay, so the entire structure is your sarcoplasmic reticulum, but the ends of the sarcoplasmic reticula are what we refer to as the terminal cisternae. Okay, so let me just kind of draw that in over here, that these ends, okay, actually, um, so ends of sarcoplasmic reticulum. These are going to be your terminal cisternae. Terminal cisternae. Okay, so let me just kind of draw that in over here. So this would be a terminal cisternae over here, and then this would be one over here. Okay, and what's going to happen is that what you'll see is the way in which it lines up is that these two terminal cisternae, sorry, I actually spelled that incorrectly. I was just trying to draw really small. Okay, cisternae, I was missing an E, N, okay, is when you take these two terminal cisternae of adjacent sarcoplasmic reticula, and you end up with this T-tubule in the middle. Okay, that's the way it's actually structured. What's going to happen is that we collectively call that a triad. Okay, so let's just note that over here is that this particular region over here in black, okay, that I'm trying to draw here, okay, I'm just going to draw an arrow up this way over here, okay. So two terminal cisternae, so two terminal cisternae, okay, plus a T-tubule intervening in the middle, okay, it's a T-tubule, okay, that represents a triad. Okay, that's very important because what's going to happen is that something's going to happen in the T-tubules that is going to act on the terminal cisternae of the sarcoplasmic reticulum, and what we're looking for is the release of calcium. Okay, so we're going to return to this image and we're going to add on some more, but what we're going to do is move back to the PowerPoint slides and kind of catch up on some of these topics that we just introduced when drawing it out, and then adding on some more nuance to it. Coming back to slide 9, so again, we started drawing out this the skeletal myocyte together, but this is really a variation and adaptation of this particular image over here where my cursor is. But you can see that it's a little bit more difficult to unpack because the diagram is a little bit more complex. But what we're seeing is that they're trying to show you kind of a small kind of section of a particular skeletal myocyte. And these are structures that you should now already be able to identify. Okay, so again, we noted that the plasma membrane of the skeletal myocyte is going to be known as the sarcolemma. So they're showing you this is going to be the sarcolemma over here that they've kind of cut into so you can reveal the inner contents. And you can see that you have these small little holes, okay, that are found on the surface, okay, of the sarcolemma, okay, and what these are representing are the invaginations of the sarcolemma to form those transverse T-tubules, and those are going to be evidenced by this network of yellow tubes over here, okay, and you see how they're going to invest very, very deep down into the myocyte, okay, so really they're creating, you know, these T-tubules, because they are invaginations of the sarcolemma, they're going to be filled with extracellular fluid. Okay, that's going to be significant a little bit later on. A couple other things you should be recognizing in particular. I didn't draw it in the other diagram. Maybe we can add it in when we move back. But you see that we have lots of mitochondria that are kind of interspersed between these myofibrils. Because again, remember, in order to create a contractile and a tensile force, that is going to require a fair amount of ATP. Okay, so lots of mitochondria are present. As well, we have lots of we have a multinucleated cell. So they're showing you kind of one nucleus over here. This is where I probably would have changed the diagram because again the nucleus is inside and deep to the sarcolemma. But again, they did show you a nucleus there. Okay. And of course, they're kind of demonstrating, as we previously drew, that the majority of the inside of a myocyte is going to be filled with these very, very long cylindrical protein threads, which we call myofibrils. And myofibrils are going to consist of overlapping actin and myosin myofilaments. And that's what's responsible for this striated pattern of light and dark bands. Okay. Now, I noted that we have the cytoplasm of the sarcoplasm. Oh, sorry, this. Sorry. So the cytoplasm of a myocyte is known as the sarcoplasm, and we have some very important components that are found within the skeletal myocyte. And the reason why I include them here is just to inform you just how energy expensive it is in order to contract a myocyte. Okay, so again, we have glycogen, we have myoglobin, and creatine phosphate. Okay, so the first thing that we talk about is glycogen, and glycogen is the storage form of glucose. So when the cell specific cells are going to take up glucose. The majority of the time, cells just like to use up glucose as it comes, right? But there are certain cells, namely myocytes and hepatocytes, the liver cells, and what they can do is they can take glucose and they can start to form these chains. Okay, so glucose is able to be stored in a certain capacity, and if glucose is required, we can start breaking down those glycogen chains to release glucose, and that glucose can then be used to make ATP. Okay, so because we require lots of ATP for contraction of a myocyte, that's the reason why we have these glycogen stores. So lots of glycogen found within the sarcoplasm. We also have a specific protein which we call myoglobin, okay, which is very similar in structure and function to hemoglobin that's found within our red blood cells. But when we talk about hemoglobin, we're talking about kind of four subunits. Each of these subunits can kind of hold a molecule of oxygen. But for myoglobin, this is only just a single protein and can bind a single oxygen molecule. Okay, so what's going to happen is that myoglobin, okay, found within the sarcoplasm, what it's going to do is that it can hold on to oxygen, and if oxygen is required by the mitochondria, it can kind of release that oxygen and supply it to the mitochondria. Okay, so it's one of those things where the mitochondria are kind of constantly producing ATP. If we need some additional glucose, we can break down some glycogen. And by the same token, if we require some oxygen, because remember, oxygen is the ultimate acceptor of electrons, okay, that creates water, okay, as part of the cellular respiration process, okay. So again, if we need those additional components, we can gain access to them very quickly because of glycogen and myoglobin. And then the last one over here is creatine phosphate, okay, which is found in myocytes. And what's happening is that, yes, we have the mitochondria that are producing lots and lots of ATP, but if for some reason, maybe we run out of glucose or we run out of oxygen temporarily, what's going to happen is that we still need ATP to be produced. So what happens is that we have a special substance which we call creatine phosphate. Okay, and what's going to happen is that it has the ability to transfer a phosphate. Okay, so that's why they call it creatine phosphate. It can transfer that phosphate to ADP to reconstitute ATP. Okay, so again, there's a finite amount that we can create in terms of ATP. But again, what we're noting is that, again, it's very energy expensive to contract and create a tension force in our myocyte. So therefore, we need to find ways to maximize the amount of ATP that we can create, and in times of scarcity, how we can kind of create some extra ATP if needed. Okay. We drew in our diagram, we drew them as these kind of red threads, but these are going to be our myofibrils. These are going to be our contractile fibers, and they're showing you in the image, these are going to be kind of your myofibrils over here, okay? And they're going to extend the length of the myocyte, and as we noted, they are going to consist of these overlapping myofilaments, okay? The actin and the myosin. So the actin would be the thin myofilament, and the myosin would be the thick myofilament. Okay, so let me actually just kind of draw that in. So remember, actin is going to be the thin, okay, myofilament, and then this is going to be the thick myofilament. Sorry, that doesn't look like there's thick right over here. Okay, and we noted that surrounding the myofibrils, okay, you have multiple sarcoplasmic reticula, and those are evidenced kind of by these blue, they kind of, well, to me, that's why I describe them as water wings that are kind of surrounding the myofibril, okay, or the portion of the myofibril. And again, this is going to be a storage for calcium, and calcium is going to be very important in initiating the contraction of the myocyte, okay. And again, we also noted that the ends of the sarcoplasmic reticulum are what we call the terminal cisternae over here, and they're going to sandwich in between a single T-tubule over here. So that's how we kind of align it. The T-tubule in the middle, the terminal cisternae on the side, and we collectively call that a triad. I wanted to return to our drawing just for a second, just to kind of highlight what we're going to talk about next. So remember, we looked at these myofibrils, these very, very long threads that are running the length of the skeletomyocyte, and they consist of these overlapping actin and myosin myofilaments. So again, if you look at the third one down, I drew a small little box and I said, let's kind of zoom in on one particular part of this myofibril. And what we're viewing in particular is that we have these actin and myosin myofilaments that are overlapping each other. Okay, and what we're going to look at in particular is what are going to be the structures of those particular myofilaments. So again, I just drew them as kind of straight lines. Okay, but in actual fact, there's some very important structure you need to be aware of. So let's kind of flip back to our PowerPoint slide together. Okay, let's have a closer look at the structure of these myofilaments. So we're going to start off with myosin first. So the thick myosin myofilament consists of a collection of myosin proteins. Okay, but what's very interesting about these myosin proteins is that, first of all, let's kind of unpack what they look like. So down here below, so they're showing you this is going to be a myosin myofilament. Okay, but it's going to consist of lots of myosin proteins. Okay. And when we're talking about the individual myosin protein, it actually consists of two peptide chains that are kind of twisted around each other. Okay, and so if you're kind of looking over here, it kind of looks like two golf clubs that you've kind of twisted around, around, and around, and only the clubs are present over here. So you can see over here that we have the myosin tail over here, okay, and we have the myosin heads, okay. The myosin heads are significant because these are considered ATPases. So they have the ability to kind of cleave ATP to produce energy, okay, as well as ADP and inorganic phosphate, okay. And ultimately that energy is going to be required to form those cross bridges between actin and myosin. But if you have a look over here, so we're looking, this is going to be that individual thick myofilament, okay, that we saw in the previous diagram over here. But again, what's going to happen is that all of your individual myosins are going to be arranged in a very specific way. So what's going to happen is that all the tails, so these are your myosin tails over here, they're all going to line up in the very, very center of your myosin myofilament over here. So see, here are your tails, and then the heads expand out to one side, and as well you have myosin heads that are expanding out to the other side as well. So what that's implying is that eventually when we have the formation of those cross bridges, okay, what's going to happen is that myosin is going to kind of bind to actin, and it's going to end up pulling it towards the center of your myosin myofilament. Okay, so just kind of keep that in mind, that the organization of these myosin proteins is very important. Okay. Now, while myosin myofilaments consist of generally the myosin proteins all put together, what's happening is that the thin myofilament, actin, actually consists of three different proteins. So it consists of actin, okay? So actin is going to be this one over here that kind of looks like, these little kind of balls over here, okay? They actually kind of look like olives. It kind of spoke to me right now. They look like olives. And you also have some other proteins that are helping to form this myofilament, and we have troponin, which is this one over here, okay? And we also have tropomyosin, which are going to be these very long brown threads over here, brown in the image, okay? Now, actin, the little olive ones over here, the little pimento thing they're kind of showing you over here, that's representing the binding sites for myosin, okay? And so what's going to happen is that, you have all these multiple actin proteins over here. You have all these myosin binding sites. However, when the cell is at rest, these binding sites to myosin are blocked by tropomyosin. Okay, so remember, tropomyosin are these long threads over here. Okay, and as well, tropomyosin is kind of anchored in place by troponin. Okay, so in order for us to form these cross bridges between actin and myosin, something needs to happen with troponin. Okay, so what's going to happen is that troponin is actually going to be sensitive to calcium. Okay, and once calcium binds to troponin, okay, so let me just kind of draw a calcium. Ooh, sorry about that. So once we have calcium, okay, it binds to troponin over here. it's going to cause a conformational change that's going to push tropomyosin aside, and it's going to reveal those myosin binding sites on actin, and that is what's going to help facilitate the cross bridge. Okay, so you see we have all these different kind of parts that are kind of coming together. And again, we've already previously noted that where's this calcium going to come from? It's going to ultimately come from the sarcoplasmic reticulum. Up to this point of the lecture, I've introduced myofibrils as consisting of collections of overlapping actin and myosin myofilaments that are kind of lined up end to end to end to end. But now we're going to assign an official name to that. So when we talk about the functional unit of a myofibril, we're talking about the sarcomere. Okay, and so you can see that what's happening is that a sarcomere is going to consist of this discrete overlapping arrangement of thick and thin myofilaments that kind of repeats as we kind of move from end to end of a given myofibril. So I just want you to kind of visualize in your head, visualize this myofibril, this very, very long thread. Okay, and if we zoomed in, we can definitely see that we have actin and myosin overlapping each other in a sequence. Okay, but if we kind of looked at the repeating sequence, that would constitute your sarcomere. Okay. So again, a myofibril is going to consist of many, many, many sarcomeres that are lined up end to end. Okay, so again, let's just kind of look at some of these structures together and review them. Okay, so we noted that we have the presence, if we're looking at a particular sarcomere, we're looking at actin and myosin. Okay, so again, here's your thick filament, that's going to be myosin. And again, we can see that we have that structure where we have all those myosin proteins where the tails are aligned at the middle, okay, of the sarcomere, which we call the M line. Okay, and then you have all the myosin heads that are kind of moved off to the side. Okay. And then we also have our thick filaments, our actin over here, okay, and we know actin, our thin myofilaments are going to consist of actin, troponin, and tropomyosin. Okay. Now the ends of our sarcomere over here are what we refer to as the Z disc. So this is the Z disc over here, okay, and on either side, so the Z disc is going to bookend the sarcomere. There are a series of proteins. You don't need to know their names, but they are responsible for anchoring those actin, those thin myofilaments. Okay, so that's going to be your Z disc. So that's going to be your delineating boundary for your sarcomere. Okay, and then of course we started to look, and again we introduced in our diagram that we have these different bands. Okay, so what's happening is first of all we have our A band in the middle. Okay, and our A band is going to span the width of our myosin, our thick myofilaments over here. Okay. So this is what's going to constitute the dark band of that striate appearance when we look at it again. So this is going to be your A band, that's the dark band, and then we also have our light band, which is going to be the I band, okay? And this is the areas where we just have actin. So you can see that you have some actin from this particular sarcomere over here, okay? That's forming part of the I band, but as well, remember what's happening is that you also have the actin myofilaments that are going to be a part of the next sarcomere in the sequence. So what's going to happen here is that this is going to be the second half of the I band. Okay, so A band is just myosin, and then I band is just actin. Okay, we also have an additional zone which we call the H zone. Okay, so this is going to be this region where we just have myosin. Okay, so we just have myosin over here. Okay, and there's no actin present over here. But you can imagine that as the sarcomere is going to shorten, as actin and myosin overlap to a greater degree, what's going to happen is that this H zone is largely going to decrease in size. Okay, so you should kind of be familiar with these different parts of the sarcomere and what they represent. Let's add a little bit more to this image over here now that we know about the sarcomere and we've kind of assigned a name to it. So we noted that the sarcomere is going to be the functional unit of the myofibril. So what I'm going to do is I'm just going to kind of draw in, okay, and I'm just going to say, okay, here's going to be a sarcomere, okay, and of course there's going to be another sarcomere over here, okay, and another one over here. And we know that those sarcomeres, we line them up end to end, they consist of overlapping actin and myosin myofilaments. But the thing is, remember, you have lots of myofibrils, okay? And so we kind of, if we look and the way in which all these myofibrils are aligned, all of their sarcomeres are going to be aligned as well. So I'm actually just going to even draw it up here, okay? Um, and I'm just going to denote that we also have sarcomeres over here. Okay, so all the sarcomeres are lined up end to end in a given myofibril, okay? But as well, across myofibrils, all the sarcomeres are aligned, okay? And so what's going to happen, okay, and much like we described down below over here, that if all the sarcomeres are going to line up in particular, okay, then what's going to happen is that because we have all these thick myofilaments, okay, that are aligned up together, okay, across the different myofibrils, okay, what's going to happen is we're going to end up with that characteristic dark band, which we call the A band, okay, and then we have all of our actin myofilaments, okay, all in the same location, okay, because all the sarcomeres are aligned across the myofibrils, and that's going to give us our light band, our I band, okay. So let's just kind of move over and let's kind of revisit that slide from the beginning over here, okay. So we're looking at why we have the alternating light and dark bands that comprise our striated appearance. Returning to an image that I introduced earlier on in the lecture, now we have all the information that's required to unpack what we're seeing in these particular histology images. Okay, so again, we previously introduced that this is a section of a myocyte. So where my red cursor is, there's a section of a myocyte, okay, a skeletal myocyte, multinucleated. Okay, so we have nuclei off to the side because again, the bulk of the sarcoplasm is going to be taken up by all of these thread-like structures, these myofibrils, okay? And all these myofibrils contain actin and myosin, okay, arranged in sarcomeres, okay? And because these myofibrils are all aligned with each other, so their sarcomeres are aligned, okay, what happens is that now we end up getting that very characteristic banded appearance, okay? And so you can see over here, if we're looking at this electron micrograph, okay, now we can start to identify these different regions of the sarcomere, okay? But again, you need to kind of picture that, when we're looking at, this entire portion over here, what we're looking at is we're looking at tons and tons of myofibrils and how they are kind of contributing collectively to this striated appearance. So we're seeing over here, okay, is that the dark band, okay, so these dark bands over here, okay, those are going to be the A band, okay, so that's the A band, okay. The light portions over here, okay, here, here, and here, okay, those are going to be the I band, okay, where it's just actin, okay, and then right in the center we have the Z disc, okay, so we can kind of note, if I kind of just draw in over here, this arrow over here and that arrow over here, Z disc, okay. And in particular, I also wanted to kind of note the MI stands for mitochondria. Okay, so there's going to be your mitochondria over here, lots of mitochondria. Okay, and then the N is going to be the nucleus on the side. Okay, so everything is aligned, which is wonderful. Okay, myofibrils are aligned, sarcomeres are aligned. That's what gives us our striated appearance. But again, now we have to talk about what's happening, right? Because remember, we have a degree of overlap between actin and myosin. And what's going to happen is that in order for us to create some sort of a tension force in our myocyte, we need to increase the degree of overlap between actin and myosin. So what's going to happen is that if we allow for the formation of cross bridges between actin and myosin, what we're going to do is that myosin is going to bind to actin and pull those actin myofilaments closer towards the center of the sarcomere. Okay, so they're kind of noting over here. So they're showing you kind of two sarcomeres. Okay, so we kind of even note where they show you the Z discs. Okay, so these are going to be the boundaries for your sarcomeres. Okay, and what's going to happen is that when we start to form these actin-myosin cross bridges, okay, what's going to happen is that we have actin being brought closer and closer to the midline of our sarcomere. So that's going to be our M line. Okay, same thing goes over here. Okay, and depending on how many cross bridges we form, we can actually create a greater degree of tension force. So they're showing you kind of the relaxed muscle in the first one over here, okay? We have the partially contracted muscle, okay? And then we have the maximal contracted muscle over here, okay? So we're thinking we need to activate all these sarcomeres and all these myofibrils at once, okay? And when we all contract at the same time, ultimately that's going to be the contraction of the muscle as a whole. All right, now what we're doing is we're approaching the end of our lecture where we're going to bring everything together. So we've kind of named all the players, we've talked about all the different structures, we talked about how in order for us to create a tension force, we need to increase the degree of overlap between actin and myosin in our individual sarcomeres and all of the sarcomeres that are found within the myofibrils of our myocyte. So let's bring everything together and let's talk about how we make that happen. How do we facilitate this formation of actin-myosin cross bridges? And so this is going to be this concept of skeletal muscle excitation contraction coupling. Okay, so how do we couple, how do we bring together an electrical event that's going to stimulate the contraction event? Now we noted that skeletal muscle is under voluntary control. So what happens is that these skeletal muscles are innervated by somatic motor neurons, okay, that are going to be transmitting action potentials from the central nervous system, okay, providing instructions for these muscles to contract. So again, what's happening is you have all these somatic motor neurons that are heading towards these different skeletal muscles, okay? And for a particular skeletal muscle, what's going to happen is that you're going to have a somatic motor neuron that's going to kind of divide up into a variety of axon terminals. So these are kind of the terminal end of your neurons, okay? And these axon terminals are going to be up against and very closely associated with myocytes, the skeletal myocytes, okay? So you think about it that you're going to have nerves, okay? So bundles of neurons that are heading towards specific skeletal muscles, okay? And within those nerves, okay, you have lots of these neurons, okay? And each of these individual somatic motor neurons are heading towards specific regions of the skeletal muscle, and then their axon terminals are going to become closely associated with individual skeletal myocytes. So from there, that point of contact, so you can see that here's a somatic motor neuron, okay, and you see you have these different axon terminals, the ends of your neurons. They're going to closely contact the sarcolemma of a given skeletal myocyte, and from there that's going to form what we refer to as the neuromuscular junction, okay? And so as we have this electrical signal that's kind of coming down from this somatic motor neuron over here, as that electrical signal kind of is passed to these axon terminals, what's going to happen is that there are going to be a series of events. And what's going to happen is that we're actually going to have an influx of calcium that enters into the axon terminal of these motor neurons, okay? And in doing so, that's going to cause the release of neurotransmitters. So these are going to be chemicals that are going to be released from the axon terminals. So you can see over here, they're kind of demonstrating it as these reddish portions over here. So in this case, it's going to be the neurotransmitter acetylcholine, okay? And from there, those neurotransmitters are going to bind to ligand-gated ion channels on the sarcolemma. So that's what they're showing you kind of here in green, okay? So if we kind of zoom in over here on this particular portion of the diagram over here, what we're seeing is that we have the release of neurotransmitters like acetylcholine, and what they're going to do is that they're going to pass across the synaptic cleft, and they're going to bind to these ligand-gated ion channels. And again, when we talk about ligand-gated ion channels, we're referring to channels that can open and close, okay? But it's only contingent on a particular ligand, a particular substance that's binding to this channel. And once that ligand, that neurotransmitter, binds to that channel, we have the opening of that channel, and then we have the influx of sodium, okay, as evidenced by the green, kind of entering into the myocyte, okay? And that is particularly significant because if we have sodium, which is a positive charge ion that's entering into the sarcoplasm, what that's doing is that's changing the electrical environment inside the cell, okay? And in doing so, that is going to be the stimulus, which is going to initiate the skeletal muscle action potential, okay? So we're going to actually create an electrical signal that is going to be propagated along the surface of the sarcolemma. Okay, So let's have a look at that over here. So what I want you to see over here is that they're showing you this is kind of a very um, simplified version of a portion of the myocyte. So they're showing you in blue, this is going to be the sarcolemma over here, okay? These dips down here are going to be the T-tubules, okay? And then of course you have your sarcoplasmic reticulum, okay, in yellow over here, okay? And so what they're demonstrating in particular is that when we generate this skeletal muscle action potential, okay, it's going to propagate along the surface of the sarcolemma, but it's also going to dip deep down into these T-tubules over here. Okay, so you can see they're showing you the kind of the action potential is going to be the signal that kind of runs along the sarcolemma and dips deep down here. Okay, it will probably kind of come up again and then go back down again, like so. And what's going to happen is that as that signal kind of passes by the terminal cisternae, okay, of the sarcoplasmic reticulum, it's going to cause the release of calcium, okay? And calcium is going to trickle down, okay, and it's going to, because these myofibrils are so closely associated with the sarcoplasmic reticulum, calcium is going to bind to troponin, okay? It's going to push tropomyosin aside, and it's going to expose those binding sites on actin for myosin. Okay, so this is where we're kind of start to facilitate how cross-bridge formation takes place. Okay, so let's kind of put a hold on here. I just want to kind of draw that into our diagram to finish it off. Hold on for a second. Okay, so I'm going to draw and just kind of draw in the neuronal portion over here. So I'm actually going to just choose a new color. I've been using teal quite a bit. Let's use that one over here. Okay. So what we're going to draw over here is I'm going to draw the axon terminal of a somatic motor neuron. Okay, so it's going to kind of come down like so. Okay, and it's going to contact the surface of the sarcolemma. Okay, so this is going to be the axon terminal. Okay, of somatic motor neuron. Okay. And again, the point of contact between the axon terminal and the sarcolemma, okay, which I'm going to denote over here, okay, this is going to be your neuromuscular junction. Okay, so neuromuscular junction. Okay, and again, remember what's going to happen is that we have the release of neurotransmitters. Okay, so we have the release of neurotransmitters, okay, that are going to bind to ligand-gated ion channels on the sarcolemma surface, okay, and from there we cause the influx of sodium, okay. So let's just, I'll just put this in as the release of neurotransmitters, okay, that occurs, okay. So again, remember you have the action potential kind of coming down, so action potential, uh, propagates towards axon terminal. Okay, we have the release of neurotransmitters, and then from there, if we have that huge influx of sodium, that's going to depolarize and change the internal cellular environment of this myocyte. Okay, and that is going to be the stimulus that's required to generate the skeletal muscle action potential. So I'm going to actually demonstrate that as an orange over here. Okay, so I'm just going to demonstrate over here. And this electrical signal is then going to propagate along the surface of the sarcolemma and then deep down into the T-tubules. So this is going to be your skeletal muscle action potential. Okay, and you see it kind of goes down, it goes up, it goes across. Same thing goes down and goes up as well. Okay, and what's going to happen is that as it passes by those terminal cisternae, okay, we're going to get the release of calcium. Okay, so again, I'm going to show you over here that we have the release of calcium that takes place. Okay, so release of calcium. Okay, release of calcium. And because we have all those myofibrils, you know, remember the sarcoplasmic reticulum is surrounding the myofibrils, the moment you release the calcium is going to kind of settle onto the myofibrils, okay? And what we noted is that calcium is going to bind to troponin, okay, on the actin myofilament. Troponin moves tropomyosin aside, and then we expose the binding sites for myosin on actin. So this is what we refer to as excitation-contraction coupling, right? We have an electrical stimulus, okay, that's provided by the somatic motor neuron that generates an electrical signal, okay, the skeletal muscle action potential, which is propagated along the surface of the myocyte, dips deep down into the T-tubules and causes the release of calcium. And then, as you know, calcium is going to interact with the actin myofilament to expose the myosin binding sites on actin, okay? But again, that's only half the story because we need to actually figure out what's happening on the myosin end of it. How does myosin bind to actin, okay? And why does it require ATP? So that's this last one over here, this cross-bridge formation, because we assume that once calcium is released, the myosin binding sites on actin are available, okay? But we now need myosin to form that cross-bridge, okay? So what happens is that this is a process that requires ATP, okay? So that's the reason why we require lots of ATP, and that's why we have lots of mitochondria in our myocytes. So first of all, what's going to happen is that ATP is going to bind to the myosin head, okay? So we're going to start off with this region over here in between step 4 and step 1 over here. Okay, so they're showing you here's going to be your actin myofilament, and this is going to be your myosin myofilament over here. And we see that we have our myosin heads. And so remember, our myosin heads are ATPases, so they're going to bind ATP. And as a result, what's going to happen is that the minute the ATP binds, that's going to cause the detaching of the myosin heads from actin. Okay, so that's pretty important first. So ATP binds, and then the myosin heads are going to detach. Okay, and what's going to happen is that as an ATPase, these myosin heads are going to hydrolyze ATP, okay, and they're going to form ADP and an inorganic phosphate over here, and as a result, they become energized, okay, because again, we've released that energy because we have broken that high energy phosphate bond, okay. So the first thing that's going to happen is that once we hydrolyze ATP and release that energy, we are going to lose that phosphate, and the minute we lose that phosphate, then the myosin head is going to bind to those revealed binding sites on actin. Okay, so that's the initial formation of the cross bridge. Okay, and what's going to happen is that when ADP, okay, so remember ADP is still attached. When it is detached, okay, what's going to happen is that these myosin cross bridges are going to rotate towards the center of the sarcomere, the M line, and that's going to be what's known as our power stroke. So this is in step 3 over here. This is where we actually get the increase in the overlap. Okay, because we are kind of pulling actin over top of the myosin. Okay. And then eventually what's going to happen is that, again, this is very quick. What's going to happen is that ATP is then going to bind to the myosin head again, okay? And then we have the detaching occurring, okay? So the thing is that this is kind of a cycle that kind of continues as long as there's ATP present and as long as calcium is being released, okay? So that's the reason why as you kind of contract a myocyte and you contract a skeletal muscle in general, what's going to happen is that we keep increasing the degree of overlap because we keep forming a actin-myosin cross-bridge. Okay, we detach it, and then we create another one, a subsequent one, detach and create another one. So really what we're doing is that, as we kind of increase the degree of overlap and we keep creating more and more cross-bridges, ultimately we are going to bring actin closer and closer and closer to the M line and create the maximum amount of tension in this myocyte and in this muscle. So that is going to be cross-bridge formation. So just kind of make sure you're aware that yes, we have the release of calcium from the sarcoplasmic reticulum that exposes the binding sites on actin, but at the same token, we also have to think about the other side of it, what's happening with myosin, okay, and is there ATP available for it to form the cross bridge and also to kind of create that power stroke that will increase the degree of overlap between actin and myosin. Okay, so again, I'm including this image over here because this is going to be a kind of a good review slide and summary of all these different steps along the way that are required to contract skeletal myocytes to create this tension force. Okay, so have a look at this one over here. Kind of this is a good summary of what we've just previously covered. Now, just to kind of finish off with our key points. Okay, so as we noted, we introduced our third type of tissue, which is muscle tissue. And we noted in particular is that the majority of the muscle tissue is going to be comprised of these specific cells, which we call myocytes. Okay, and we noted in particular that skeletal myocytes are they're under voluntary control, so they're innervated by somatic motor neurons. And we also spent a lot of time discussing the reasons why they have this striated appearance. That we have this very precise organization of our myofibrils, okay, such that the sarcomeres are going to line up end to end and also are going to be aligned across the myofibrils. Okay, so that's what's going to give us our characteristic light and dark bands of our striations. Okay, now in the last half of the lecture we talked about this this concept of excitation-contraction coupling. Right, we said that we have action potentials that are transmitted via these somatic motor neurons. Okay, and from there they're going to reach these axon terminals, causing the release of neurotransmitters. And from there, those neurotransmitters are going to cause the depolarization of our sarcolemma, of our skeletal myocyte. Okay? And what's going to happen is from there, then we can propagate this electrical signal all the way along the surface of our sarcolemma. Okay? It's going to dip down into our T-tubules and then cause the release of calcium from the sarcoplasmic reticulum. And this is what we were waiting for, right? Because calcium is going to be released. It's going to bind to troponin, push tropomyosin aside, to reveal the myosin binding sites on actin. Okay, and then as long as there is ATP present, okay, what's going to happen is that myosin is then going to hydrolyze ATP, okay, form the cross bridge, okay, and then initiate that power stroke that will increase the degree of overlap between actin and myosin. So that's going to be our sliding filament mechanism. Okay, and so presumably, if we are coordinating everything, okay, and all of the sarcomeres of a given myofibril are shortening at the same time, okay, and all the myofibrils are shortening at the same time, that means that the entire skeletal myocyte is contracting at the same time, okay? And then, you know, if you kind of expand out even more, if all of these skeletal myocytes are shortening and contracting at the same time, that is really our skeletal myocyte creating this tension force collectively. Okay, so there's a lot to kind of unpack, a lot of lingo that sounds the same, myofibrils, myofilaments, but again, you know, kind of review it again, and it'll take some time, but I'm here if you need some support and need some additional clarification. So let me know if you have any questions by email, but that's all I had for today. All right, take it easy. Bye now.